So yeah, thank you AWS and Midwest for inviting me to speak today on some work that was done on us looking at how we could build a platform as a service, looking in the world of biology and genomics, and in particular in forensics. So, I see U of Michigan. I'm watching the chat here. Hopefully I don't get too distracted by whoever that troublemaker is, but we'll continue. So a little bit about me, just so you know where I'm coming from. I'm a PhD in biomedical science, and over 10 years ago, we were doing vaccine research, and a lot of that required bioinformatics because the data sets were getting large and they were more sophisticated, and for us to be at the leading edge, we had to do a lot of data analytics. And so I'm more or less a self-taught bioinformatician, and have had a chance in leading some government-sponsored programs as a contractor to turn those technologies into R&D solutions that analyze large genomic DNA data sets, specializing things at the bench in the laboratory, and then also building the software that's needed to analyze those data sets. And what you're going to see today is work that I transitioned to the University of North Carolina with. I was faculty member there before returning back here to Ohio State. So I know we don't have the ability to do questions in real time easily, but I'll try and watch the chat if something comes up, and at the end, maybe we'll have a chance to catch up on questions. So we're going to get a little bit of a background here, just so we know what we're talking about with regard to data structures and we're on the same page. So here on the screen, you see some bones. These are human remains. On the left is a scapula, shoulder type of bone, and on the right top are some finger bones. Now, in the forensics lab, it's often the case that they get these type of samples in the door from human remains that are highly degraded. And it's a challenge with some of the existing DNA technologies to get really strong, robust signals that help you identify who the person is. So what you'll hear today is about how we've been looking at new technology that gives us higher resolution, it's more sensitive, more data, in fact, but we need really good resources on the compute side to analyze that, and that's where AWS comes in. All right, so a little bit of background, and here's the obligatory Buckeyes picture. So we'll talk a little bit about genetics. Believe it or not, we're all 99.9% identical. And there are some slight differences in our genomes amongst humans, and they fall into three general categories, at least for what we'll be talking about today. The first is this funny one called single nucleotide polymorphisms. We call them SNPs. So if you want to impress your friends and family in this field, call them SNPs. We have insertions and deletions, which are exactly what they sound like. It's when the genetic code is missing pieces or has extra pieces added. And then we have copy number variants, which we'll talk about today. And we also call those STRs, or short tandem repeats. And I'll talk a little bit more on that. And the human genome is quite amazing, right? As just having 4 letters in different configurations, but over 3.2 billion of those letters spread across 23 chromosomes, right? So for us to be able to analyze the whole human genome, which we do every day now, thanks to some really great lab technology and really great workflows on the computing side, we've made major discoveries. So we're going to talk about a much more refined part of the genome, these STRs, but just bear in mind the enormity of the human genome and really what it might take to fully analyze that. And when we look at genetics and forensics, I broke these down into 4 areas. And these are just more for reference for the community, and these slides are available online for you to look back to. But we'll be talking about these short tandem repeats. We also call them microsatellites. And they're used primarily for identifying individuals. It's also the primary type of data structure it's used in databasing and forensics, and then also some paternity testing. And then these other 3 areas, again, just for reference, we also look at another DNA element called the mitochondria. We look at these SNPs I mentioned earlier, and then there's some new and evolving areas where we're looking at maybe even the whole human genome, those 3.2 billion base pairs, or other scattered regions throughout the genome to get really high density data. But that's for another talk. So on these STRs, they're really the gold standard. This technology has been around for about 20 years now in the community, and has been readily used by the forensics community for that long. And in general, the markers that we use, they're tandem repeats. So you would have 3 to 5 of these ACs, Ts, or Gs in tandem. They're in a repeat motif. And at the bottom, I try to give an illustration, because what we do is look at the length of that gene segment. You can think of it as measuring like with a ruler. So on the left, person A might have 2 copies of this genetic marker, and copy number 1 has 8 repeat motifs in it, and copy number 2 only has 5. So quite literally, when you database this individual, those elements would be 8 and 5 for that genetic marker. And the person on the right, person B, they happen to be one of these people we call homozygous for this because both of their copies are the exact same number, but we would still database them as a 6-6, right? So there's genetic identity that separates these 2 individuals, one being 8-5 and the other being 6-6. We look across 20 different markers so that we get some really great statistics and can use probability-based rubrics to understand the likelihood that samples belong to each other. But think of it more as these tandem repeats, and we measure the lengths of them, and that's what's presently done for databasing. And if we tear this apart a little bit more, you can see at the very bottom a little bit more of an illustration of those repeat blocks, where you might have AATG, AATG, and it's repeated over and over. And in this example, you see 8 in the top track and 9 in the bottom track. So this could be another example of an individual's genetic identity for that particular marker. And when we look at what's been done to date in the forensic space, back in 1994, there was federal legislation passed that established the National DNA Index System. And the overall platform that runs that's called CODIS. CODIS runs at the local level, as you see in the graph or the picture on the right, and then up through each state that manages their own part of the index, and then up to the national level, which is managed by the FBI. And the system is quite robust. There's over 19 million profiles as of April of this year. I broke those down to show you just some representation of what's in each indice. Over 14 million offenders. So this is an individual who's committed a crime and has been incarcerated. Their genetic profile is taken and put into the database for recidivism purposes. And we have close to 4 million arrestee profiles. So some states are allowing collection upon arrest. And then we have over 1 million forensic profiles. So these would be crime scene type profiles that are in the database waiting to get hit from potentially one of the arrestees or offenders. The system's been quite successful. We have near 500 million hits. There are 511 million. And then over 500,000 investigations have been aided. So this is really an investigative tool, right? It doesn't always solve the crime. It's not the data that's admitted into court, but it's used for investigative purposes to try and find individuals and connect them to crimes. And as I mentioned earlier, this technology is well over 20 years old, and it's called capillary electrophoresis. And so we thought it's time for an update. And so in comes this new technology that I'll be talking about today, which is called next generation sequencing. And I'm showing this graphic. It's a very prevalent image in the genomics community to talk about the cost of generating sequence data. So the cost of looking at all those 3.2 billion base pairs across the human genome over time. So on the x-axis, you can see that around 2007, 2008, we introduced a new technology in this space that was massively parallel. And so by this, instead of doing a single small string of DNA per time, we were doing millions, and the technology got better and better over time. So you can see now in 2019, we're at the point where we can analyze a human genome, those 3.2 billion base pairs, at close to $1,000. And most of the talk and data you'll see today, these samples are along the lines of $40 to $50 a piece, which is quite tremendous. And then I thought, there'd be some appeal to this audience to see, the linear rate of Moore's law and its decline. You can see that we went exponential with our technology improvements in the DNA space, although we might be flatlining and looking a little bit more linear and tracking with Moore's law, just a different intercept of the slope. But nonetheless, just to talk about every laboratory now, big or small, has access to this new next generation sequencing technology. So if we start to break this down, what do the data structures look like that come out of these next generation sequencers? This one that you see on the screen, it's from the company called Illumina. The machine in particular is called MiSeq. And this is the instrument that will generate the DNA data in the laboratory. And the data comes off more or less as this flat file, as you see on the right, where at the very top of the file, you can see the @ symbol, and that's just an identifier to give you information with regard to the instrument, the date, the user, some specifics about which of the millions of data strings that are coming off this, the one is in particular. And then you see below this, of course, the familiar ACs, Ts, and Gs that I worked with every day of my life for 20 years. You see a plus symbol, and that plus symbol then indicates that below there, there's some fields that are in ASCII text to represent the quality. So each one of those letters and characters tell our bioinformatics software the certainty that the above nucleotide is correct. And I'll show you the breakdown of what that looks like. So it's helpful here because we can remove any type of impurities in the data set or anything that may be of low quality and could interfere with our downstream analysis. And it's easy to bake in some really lightweight operations, even ones that we've been able to do on serverless functions through some simple Python scripts. And then I'm just showing you another read here below that, in the middle part of that black box. And then at the bottom, you see up to 25 million reads. And this is one of the smaller instruments, in fact, now that's on the market. We have instruments from the same vendor called NovaSeq, and those are producing thousands of millions of reads of data. And with that, it's nice because we don't have to just look at one individual genome from one person. We can now spool this up and start to do hundreds of individuals, if not thousands, depending on what genetic markers you're looking at. And in general, these files tend to be on the order of anywhere from 25 gigs to a terabyte or more, just depending on how many of the genetic markers you're analyzing per time. So that file format there, it was called FastQ. You'll see that extension appended to any one of the flat files that come through one of these instruments. Fast is a relic of the bioinformatic days. There was FastA originally, and then FastQ was invented, and the Q stands for quality, which you can see down below in the table. As I had mentioned earlier, those ASCII text characters describing probabilities. So on the far right, when you get to the letter J, and then up through I, H, G, F, E, D, and C, you have certainties that are greater than 1 in 1,000 accuracy, right? So we can fine tune our quality assurance parameters to say that we will only accept data that's greater than 99.99% in accuracy or lower, depending on the pipeline that you're running. So this is a slide that I use always to harass students or other people in workshops like this. That, believe it or not, there is a repeat motif up in the top track with ACs, Ts, and Gs. It's 4 letters, and then I just stopped at this point and let everyone's eyes go across and try and identify that repeat motif if possible. And then I let people shout it out. If anybody sees it, I guess type it into the chat bar. It's torturous, right? All right, so enough of that. Thank you, Kathy, that was kind. Here it is, I highlighted in red, it's AATG, and even to count those is very difficult. But, thank you for bearing with me. But, the point of that whole exercise is to say that, we can't look through those millions of lines, obviously. This whole community, no doubt, understands that, and we're already ahead of the game when I was presenting that example. But in particular in the forensic space, right, to try and teach and educate and instruct individuals that may be used to seeing histograms and other types of representation of data, this could be something that's very intimidating to think that they have to look through millions of lines of flat files. And we tell them, no, in fact, we've written smart computer software and other things that will help them with this task. And we call that bioinformatics. All right, so let's get into some of this cloud prototyping. So back in 2014, when I was at NC State University, our IT director, Chris Eichmann, was a wonderful resource for me. And I was very familiar with using, on-prem HPCs. I can't say elegantly, again, because I'm self-taught, but nonetheless, trying to work with our own our hardware and realizing at the university that the resources that were available were ones that were more optimized for some of the engineering and other computer science types of tasks and large computing they were doing and weren't really tailored for our genomics environment. So Chris had recommended that I start to play in AWS. And after the first day, I realized the value that came from that and started to work up a concept. And again, based on forensics, how could this approach be deployed in a method that would benefit the laboratory and the community at large. And this is one of the earliest concepts with this. And if you were to start, I don't know if you can see my pointer, the hand here, but in the middle, in the bottom, it says sequencer. So that would be the lab side equipment, you know, putting out these FastQ files. We drop those into some local storage on-prem, but then put that through a secure channel up into an S3 bucket. And then from the S3 bucket, right, as you're most familiar, you can encrypt that. You can make that redundant, cross-region replication, whatever you might need, depending on how large maybe that forensics lab is. You can archive that, thinking Glacier for long-term storage. And on the right of the cloud there, you know, spooling up the types of instances you need. So if we're working in Windows, because some Windows-based software, we could set up instances like that. We could also, which is what I'll show next, you know, use command line interface and set up Linux AMIs on EC2 to run some of our bioinformatic scripts that are in Perl and Python and some other languages. And then the data can come back through in a secured fashion to some sort of terminal back to the crime lab, where an operator has a chance to look at the reduced data. And then, as I mentioned earlier on the far right, that CODIS system, right, that they could keyboard enter because there's really just not a API or anything yet to interact here, but put the data up into their local, state, or national databases. And then one of the dreamy things, which is on the left-hand side here of this, is to go to the remote, you know, crime scene level analytics. And there are devices coming out that are the size of a portable thumb drive that can generate DNA data. They can plug into a laptop. So let's connect those to the cloud. Let's not have to go back to the crime lab. We could send these straight from the crime scene into the system and maybe have some central based you know, analyst that can review data that comes back. So, you know, this is my pie in the sky type of plan. Put this in front of some of my friends in the community back in 2014 and was told no way, no how. You know, we don't do this. The government doesn't use cloud. I don't think they realized at the time their email was being hosted in the cloud, but nonetheless, we were told no way, no how. But we've slowly started to convince folks, and I'm going to show you some of the work that was done in this space. So when we go back to some of the earlier work we were doing, we published a paper in 2011 in how we could do this bioinformatics over these large data sets. And in concept here, this illustration is showing you that we were trying an approach that I'll just call fuzzy string matching, right? So we're going to do some sort of, you know, loose grep type of match with a reference. So each one of these blocked out, the 3 blocked out areas would be a reference sequence or reference string that we maintain of possible identities of genetic information. And then we would take new data, which are the loose tracks of strings that you see below, and then align them so that we could determine if we had a good match. And then simply enumerate that as you see in the table to know that we had a size 5 and a size 11, or excuse me, a size 7 marker, and then how many of those did we have? And this is important then on the forensic side to do some quantitation. One of the pitfalls in this approach, for one, it was for the the algorithms we had written at the time, it was very computationally expensive to do this because of all the different possibilities and ways that we had to loop functions so many times to make sure that we're getting the best alignments in our fuzzy approach. And then a second pitfall here was that there are still sequences of the human genome that are unknown. And so when we encountered those in our validation, our V&V, we realized that there's going to be a lot of new sequences that we didn't have references for. And our algorithms were just not robust enough to hit on the new ones without, you know, risk of hitting old ones, giving us misidentities. So we had to modernize. And the next approach there was something that I'll just call tag and interpret. So what we did instead is that we knew that there were constant regions in the genome, and those are shown in the the gray boxes on your screen. And they bound the region that we're most interested in, the actual string of information we want from the genome. So by us looking for substrings and then simply splicing out what's in between the substrings and enumerating those, we could write much quicker algorithms, and they were much more precise because these gray regions were always constant and did not change over individuals. And you know, for historical purposes, we look back at the way that the lab kits were designed over 20 years ago. And similarly, because of the way that they amplify signal, they always target constant regions of the genome. So we looked for constant regions in these substrings and then simply spliced out the region we were most interested in from the superstring and then enumerated those. A colleague of mine at University of North Texas, Bruce Budoli, he and his team had written in Perl one of these approaches that does tag and interpret. So in 2014, when I started my lab at NC State, having no resource available quite yet, I just wanted to test out AWS. So I went with the straight razor, they call it, that's the name of the application here, straight razor, this Perl script, just in my first testing. And there's a link to the website if you have interest in looking at straight razor, which they've certainly modernized since the first reporting of it back in 2014. And to do this, and again, this was me just cutting my teeth on AWS. I just set up, Amazon Linux, put all the dependencies, in there, got the Perl scripts uploaded and our data sets, and then mapping to the architecture of the program, put in straight in the Linux AMI commands to execute this script. And at the end of this, it produces some structured data that I could then look at with other types of graphical interfaces and things like that. So this was a test condition right straight into a Linux AMI using Perl scripts. And it was a little overwhelming, right? And this is even back in 2015, at all the different options you had in EC2. So did we want to be on micros? Did we want compute optimized, memory optimized, IO optimized? I didn't have answers for those. So I started doing some of this testing to understand what was best for us. And given that the process is fairly linear, we observed something that maybe wouldn't have been a surprise to all of you, but it was a surprise to me. It's best illustrated if you look at the compute series, so C4 large through the C4 8X large. And you can see in gray, the bars, which are mapping to the y-axis on the left, that's a processing time for looking at 100,000 of these strings. And as you'd expect, more cores on a larger EC2 instance resulted in quicker clock time to produce our data file. However, the black line, which is mapped to the y-axis on the right, is showing the estimated cost for running 20,000 of those samples. So I figured a crime lab, moderate size might do about 20,000 of these samples a year. And those costs were about $1,000. But that was constant across all of the different compute large. And you see that too with some of the others, relatively amongst each group, the same cost. And I'm sure that the folks at AWS, when they devised their cost, per minute on each one of the EC2 instances, had some sort of algorithm that was quite linear itself. So we end up seeing the same cost. However, if I had to choose, I would say, let's go to the C4 8X large. We're going to have that done in a matter of seconds, and the costs do not change. So we could still operate at around $1,000 a year, provided we were just using EC2, on and off, that only when we needed it, we were turning those clusters on and using them. So interesting observation that we saw there with regard to cost and different configurations of the EC2. So we generally try to go with the largest one that we can afford and then be smart about utilization. And if you can see this one up on your screen now, this is showing some of that reduced data. And each line here is showing the output of each one of the genetic markers we're looking at. However, what we realized is that when we look at these data files, the laboratory equipment itself are producing errors in those strings at the rate of about 1 in 1,000 characters having an error. So when we're trying to look for exact string identification, we end up seeing, a predominance of I'll call them the correct or the actual genetic sequence, but then we end up seeing tens to hundreds for each genetic marker that are different because of the 0.01% error. And I just tried to generalize this, that if we had 0.1% errors in 5 million strings, right, we'd have 5,000 wrong strings. And we needed to find a way through some sort of statistical measure or thresholding that we could remove those incorrect data strings. Some of the analysts that we worked with, these end users, had shown us how they use in their other data analysis some histograms to represent data, because their lab side data, this older technology, also generates some information that's not quite correct. And so the way they'll handle this is to try and look at each genetic marker, which you can see here in each one of these boxes, look at the total number of reads, and then you can see a blue bar across each one of these graphs. And that's where we've, through our own validation, determined a threshold level at which we can have 99.9% accuracy. So this now can be baked into our algorithmic tools, right? That once we have reduced the data sets and we have the tens, hundreds, or thousands of reads that may be inaccurate, we can see if their enumeration values fall in the level above or below our threshold, and then remove them from final output data sets. And I have this up on GitHub. This is a RShiny application. If you guys are in RShiny, this was another fun one that I thought I'd put out in front of the group. It was new to me at the time, but the widgets that are in the user community for RShiny are very helpful for things like this when you're trying to look at lab side data in particular for us. And I put this together pretty quick to help my users and have graphical data sets, tabular data sets, and also the ability to export data all through that one application. Okay, so now we get into where we start to look at the cloud system that we were working on, those more production level. So we were fortunate that we received, when I was at NC State, funding from the Department of Justice to look at cloud computing. So we'd done enough advertising of this and talking to our friends. I guess you could say that someone somewhere thought there was hope that we could go to the cloud for forensic analysis of DNA, right? So through 2015 through 2018, we worked on this cloud system. And most of the data you're going to see is from this report here in FSI Genetics, so Forensic Science International Genetics, and how we built this cloud system. So I'll spend a little bit of time on this graphic here. Hopefully it's not too overwhelming. And remember again too that this was, well, this is now getting close to over 3 years old. So some things may be considered out of date or not best practice, but bear with me. So if we start at A1 in the bottom middle of this graphic, you can see that you have a user to terminal, and they would have one of these FastQ files likely. We also had another workflow where you could circumvent the user if your lab equipment had the right pointers to drop data into S3 buckets. We could do that too. It's just that we had, and again, this goes to end user requirements and forensics, the end user and their desire to see the data, or at least know that they're uploading it. And so we went with that model. So they would be uploading through a GUI that we developed, their FastQ file or files. And then also with that, some metadata. And the metadata would be information such as the laboratory that's submitting, the individual user that's submitting the lab equipment that was used, other information with regard to if they knew if the individual was male or female, location information. And we use all this then to create entries in our SQL database, right? So all that metadata would go and live there and create record and the keys that are necessary to track the files as they move through the system. So the FastQ file then gets dropped to an S3 for upload. And then at the time, we're using Elastic Beanstalk. And so the first scaling that we would do here is that we had a listener sitting in front of this, right? And then once data was received into the S3 bucket, kicked off appropriate to load the number of instances we need in the Beanstalk to look at the file to determine if it was in fact the correct type of file. We wanted to prevent any type of DNS attacks or any type of malicious thing, or even if just the user had the wrong type of data file. So it's verifying that it is a FastQ or even FastA file. And then if it's not, it's sent over to the left. You see in the decision selector there to a reject S3 bucket. And then we use Lambda to just send email rejection notices to that user that uploaded to say that the data file was not correct and was not accepted. If it was successful in passing through and was approved by our system, two things would happen. We'd set it up for staging in a separate S3, right? So as part of our security plan. And then moving over to the left, you can see we'd archive that same original file just so we had the original record of file. As we move up to the top of the diagram, you can see now we are reducing the data from a FastQ to a FastA just to, there's just an heuristic that we employed to speed up the process because we didn't need that quality data through the whole pipeline. We do it once, and once we understand the quality, drop that information and keep it as a FastA file. And then we've got another set of elastic beanstalks, and these are the ones that are in the core of that application I described earlier with the substring search and reporting out of the splices that we do and enumeration. That data from there can be put into the SQL record for that file. And then as we spin around the right side of this diagram, you can see that we do some destaging of the data. We're using another elastic beanstalk to complete the process to write results to file, which would be our reduced data set. Send that data set to the user, again, using Lambda to notify them that data is ready, but then also the same data going from step 9 back into the database to complete the whole operation. So this was how we had implemented this, the first version out here on Flow. This is pretty much what I just talked about, but again, since these slides will be made available to you, maybe it'll help a little bit of understanding because I went through that kind of fast. And so now if we take a look at some performance here, you can see that where in the top graph, we looked at how many minutes did it take the application to run and looking against file size on the y-axis. And this is where we were able to scale across single file up through, I think we tested up to 300 files at once. What was interesting is we had a mean processing time, about 10,600 kilobases per minute across numerous file sizes. And I know that for some of you that, think in regard to processing speed, it sounds kind of slow, I'll admit, but it's just the way that the application was running at the time, right? To try and do that bioinformatics, which I made sound very simple. There's a lot of other biological considerations that have to be taken into account, including the fact that DNA is a molecule that has 2 copies of itself. They're in polar different orientations, one going forward, one going backwards. So once we go through the file, we actually have to go back and reanalyze it in reverse, believe it or not. So there's actually a lot of computation happening at the time. What was nice, at least, is the take-home message with this is that using elastic beanstalk, we could maintain a relatively similar time of processing, no matter the file numbers that we're uploading into the system. And then in the bottom, you can just see another representation of looking at the kilobases per minute per file against the numbers of files in batch. And you can see that from 2 to 26, 60, 66, 70, and 75 files at a time, relatively similar speeds in processing. The forensics community, the security is probably the biggest sticking point for them, of course, right? And so at the time, we followed AWS guidance with the shared security policy. They're going to provide resources, and you need to make sure you configure those resources appropriately. And so the things that we implemented at the time included IAM controls, right? We found this very valuable at later stages where we had collaborators, even those from different countries, where we could define user groups and also IAMs for those individuals, so they had access only to their own data. And if there were any agreements for sharing, that we could manage those users and groups appropriately according to the data sharing policies. We also use multi-factor authentication, of course. This was root access to the cloud, and then we were using tokens for secondary authentication for users. We put all this inside VPC. We found that to be very helpful. The university was also working on its own cloud policies at the time, so we were helping to define those for them, and we were able to implement VPCs and DNS that were both compliant with our system, but then also the university's requirements at the time. We encrypted data, AES-256 for the S3 part of the system, and then we also, of course, did encryption of data in transit. TLS was popular for most of what we were doing. And then we looked at RD Gateway for the Windows application, and then SSL for Linux. This slide here, just wanted to show you, remember early on I said it'd be kind of a dream to go to the crime scene and be able to analyze a data set. The picture on the left is that device. It's called a Minion. It's made by a company in Oxford. England, and we were able to put samples on this, generate DNA data. It's not in the FastQ file format. Originally, they used this format called HDF5, so we had to write a little lightweight script to convert that to FastQ before admitting it to our system, but we could actually do that, connect to the cloud, and generate data, which you see on the far right in the red box. The numbers, if you try and tease this apart, you'll see that we were getting 10 to 20. We were getting very low levels, less than 100 of individual sequence reads, whereas those big boy equipment, right, that like vendors such as Illumina sell for the laboratory, give you thousands and hundreds of thousands of reads. We were getting low numbers, but yet they were still concordant with the correct data, which was nice. So we demonstrated this 3 years ago, the opportunity to go remote to the field. Hopefully people start to adopt this, and I understand that there's additional funding waiting to help other research groups look at this further. So, I mentioned the original version. I said this a couple times with the platform as a service that I showed you, and we're using Elastic Beanstalks. We encountered some issues with those. One was that to have a Beanstalk continuously on was expensive, right? Sitting there listening, for example, and waiting for something at S3 for the users that we had, right? Because this isn't yet a full-fledged system that's being used by multiple users. It was just my laboratory. So we're incurring a lot of expenses in keeping EBs on. We also had some issues, and likely they've been fixed by now, but at least with our large data sets, there were some issues, some timeout issues. We couldn't quite uncover the exact root cause of failure, but we thought it was something to do with the relational database and some sizes of files, and there was just timeouts. And the unfortunate thing was that because the way the data was entered into the database, we didn't know if a file had in fact failed early. We just hadn't, produced the system that way to notify us of that. So we may have partial data sets in there without knowing it. We just happened to stumble across some things that looked odd to us. We had to investigate and found that to be the case. So what was our solution? We worked with the AWS architects that were assigned to us at NC State. Ben Snively was one of those, very helpful for us. And recommendation was made for us to go to Dockers, right? And so, of course, this is becoming quite norm for doing analysis like this. But we moved to this back in 2017. So we removed all the Elastic Beanstalks and went to spot instance of Dockers. Then we also bumped up the size of the RDS, right, to overcome any type of memory issues that we were having. The Dockers worked quite well for us. It sped up operations, and we didn't have any failures from that point. And of course, the costs were much lower for us to be using spot instance Dockers for this type of application versus the Elastic Beanstalks. So I've already said most of this, but when we looked at, what was really great, the security was awesome. Undoubtedly, we can, go to folks in the forensics community and tell them how secure these systems can be. The data sharing was a really great benefit of this. We were collaborating with people throughout the US and abroad. We love the scaling and demand of this. And we also liked being able to build user interfaces to end user requirements. And that was something that helped us. And most people in design know this, but by us going to the forensic practitioners first and asking what they wanted for their system and building to that really helped us do a great job. And I told you we switched to Dockers, and we had still some community aversion here, but we're working on it. And then, unfortunately, the funding has run out. So if anyone's really wanting to, try this out, unfortunately, the system has been packed up and images made, and it's not active right now because the NIJ funding was only for 3 years. If you have a chance to look at the slide deck, you can see some information about, I want to brag, the Ohio Crime Lab, Bureau of Criminal Investigation. While I was at a company called Battelle here in Columbus, Ohio, we helped transition some of the technology I showed you today, not the cloud, mind you, but their on-prem analysis to the crime lab. And they're now live this summer. They're one of the first 3 laboratories in the whole country to use this next generation sequencing technology I talked about today. And they're using this for missing persons casework. And importantly, here in Ohio, there's roughly 1,000 missing individuals, and most of those are children, right? So they're doing important work in that crime lab. They're using high-tech technology and next-gen sequencing. Now they're not cloud yet, but who knows what's going to hold for the future for them. I just want to give a shout out there. And then in a matter of saving a little bit time for questions. Just get to my thank you slide. A lot of this work, as I mentioned, was done at NC State. Wonderful team there, all listed. Ohio Crime Lab provided amazing support and help through parts of the program, as well as some of the private companies, including Promega, Illumina, Ion Torrent, Battelle, and then we had folks from NIST as well helping us on the program. So I will leave it there and let you guys ask questions.
Wow, that was amazing. Hopefully we got some questions. If anyone has one, please place them in the chat or in the Q&A. There was, excuse me, one comment earlier. You got a +1 for Shiny.
Go Shiny.
All right, everyone, we've got a couple minutes left here, so if you do have a question, get it in. I do have one, Seth, which is I was fascinated by the fact that you were, you mentioned a couple of times that you're self-taught.
Yeah.
Any, you know, advice to people who find themselves in the same situation? How did you come up to speed so quickly?
Oh, it's amazing, you know, necessity is mother of invention, right? So I had a computer and my mission was to be able to analyze, you know, those forensic data sets. And so, you know, I was up late just looking at videos online, reading things, whatever documentation was out there, asking my friends. So I had a lot of really smart people in this space that were kind when they look at my scripts and say, well, you're missing a comma here, you know, type of thing. So being humble, being diligent, I guess, and then just having some sort of problem you're trying to solve. I think going into this maybe without that problem and just wanting to learn, I don't think for myself I would have gotten as far.
We do have a question here. Adam was asking, and I'm bound to mispronounce this. I was curious if you had experienced using, is it Motif for sequencing?
Yeah, Motif. That is something I never got into. I know that some of my colleagues use that, even those at OSU. There's a competing technology called QIME, QIIME, and I have more familiarity with that than I do Motif, but I know Motif to be a good one. And I can follow up offline with Adam if you'd like to talk more.
Any other questions for Seth? Not to be, yes, that's the first thing I thought of too. Not to be confused with AWS Chime.
Good one, Thomas.
All right, great. Well, thank you so much. That was just absolutely fascinating, and I know my head is spinning in a good way. So appreciate you taking time.
Great. Thanks, everyone.